lab there at Virginia. She's not with us today. She's actually in Portland, so she's also. My project on the fitness of the American bellflower in a novel environment. So these are all the populations that we have up here at PCCI. In Virginia, you can see that kind of little golden star. That's where she's at, and all of the plants that we have up here were grown from seed in the common gardens there. And once they were big enough, they brought them up here, and we planted them all straight that way. So every single one of those populations, so we got Florida, Georgia, Kentucky, Arkansas, Kansas, all over the United States, they're all planted up here. So we're trying to tell the difference, or trying to see if there's a difference between how they cope up here on the range edge. You can see the range edge as that black line all the way around all the populations. And there is one that's all the way down outside of there too. So my objectives for this project, first to see if reproductive phenology differs among populations. And if any of you don't know what phenology is, it's just the timing of things. So when a plant sends up a stalk or bolting, we're taking that data down to see if some plants bolt earlier than other plants. From experience, Holly has told me that plants down in like Florida will bolt a little bit later, and they'll just grow, and then finally they'll flower because they have such a long growing season. Whereas plants up here tend to have a different strategy. They'll ovulate, grow quick, flower quick because it's short. So we're looking at kind of phenological adaptations that way, because that is one way that plants are able to adapt to different environments, is alter the phenology. Secondly, we're looking at survival and fitness. So survival is pretty obvious. That's just, are these plants dead or are they alive? So we have taken a census already, plants that have died, been trampled, and eaten by slugs. And we'll take one again at the end to see what populations have fared better up here, just in terms of survival. And then also we're looking at fitness. So that's how much fruit they make, how many flowers they make, how many seeds, obviously how many babies they're able to make. And the way that we're going to be doing that is taking twist ties and tying off a section. So we'll put a twist tie down here, count 3 nodes up, put another twist tie, and that'll be our study section for each plant. And that's where we're going to be counting the flowers, and then also the mature fruits and immature fruits. And then the last one is to understand the limiting factor on plant population establishment. Essentially, if plants can move successfully and persist in a place. So does selection on phenology differ among populations? So how many days since they bolt does it take for this population to flower? Does it take 2 months? Does it take a month? We want to see those differences in populations. Like I said earlier, maybe the southern populations in the lower latitudes will take a lot longer to flower than the ones up here because of the longer growing season in the south, shorter growing season in the north. And then also we're looking at pollinator selection, so I may grab Jack over there and see if we can talk bees for a while. The main pollinator of the American bellflower is the bumblebee. They're looking more honeybees, but I suspect we may get some crossover with my plants as well. So far we have done bolting and height, and that's really all we've been able to get done so far. Planting or flowering and fruits comes later. So we've scored bolting 6 times and height 6 times, and we've been able to see if there is any variation, at least with that type of phenology, between the populations, which there hasn't been so far. So far, every single one bolted around May 22nd, and there really hasn't been any variance that we've been able to pinpoint a certain population bolting later or bolting earlier. Then slug treatments, I've been killing a lot of slugs. It's been fun. And then there's lots of growth, really. We're just waiting for them to flower. So they start off little guys, and they're getting big. Kids are growing up. That's good. They didn't all die by the slugs. So here's some preliminary data. This is kind of an obvious graph. As you can see over there, it's kind of backwards in terms of date. but the latest date over there, June 9th, is the last time we had some molting, and then we took heights on 6/13. So the plants that bolted later are obviously not as tall. That's what the back graph's saying. Over there at 16, you can see they're much higher, all the way up. The average is all the way up almost at 50 centimeters now. This is kind of a cooler graph. It's consistent height of plants along the latitudinal line. So what I did, you can see 28th parallel down there, 33, 36, 37, 40, 43. That'd be Florida, Georgia, Tennessee, Kentucky, Indiana, and Michigan. And as you can see, there is almost no variance at all in height of any of these plants. And that just tells us that the growth rates have been the same. We can't really draw any lines in terms of fitness or anything like that from that, because they're not that doesn't have anything to do with flowering or fruits produced. But it is nice to see that the growth rates so far have been equal. And then I also did longitude. So Virginia, Kentucky, over all the way to Kansas. Same thing, you can see the scale is only 24 to 26 centimeters. So there really hasn't been much variance in terms of population height that way either. This one is survival. I said we took a survival census already. And you can see that it is reduced on the range edge. So that line that I showed you on that second slide, you can see Florida down there has not survived as well, and Michigan, surprisingly, has not survived as well. My hypothesis was that Michigan would do better, because obviously it's used to this climate, it's used to less precipitation, it's used to the lower temperatures, but we have got a lot of precipitation, so that might explain why Michigan hasn't survived as well. You can see Alabama, Kentucky, and Indiana, the range center populations have done very well at about 95% survival rate. So moving forward, plants will flower in about 2 to 3 weeks, and that's when stuff gets pretty hectic. And we'll be out there every day taking pollinator observations once they do open up. And how that works is we're probably looking at about 5 plants at a time. I'll have to figure out how much I can handle, how many plants I can see at once. That's my guess. And what we do is we have a stopwatch, and for a set amount of time, we watch and see how many pollinators visit each plant. Record that number and also what was the pollinator down to genus. So that's why we might be getting some help. Record weekly flower counts and fruit maturation. So that'll happen 3 days a week, and we'll go out there, see if a new flower is opened up in our study section. Record that. And then also fruit maturation. So immature fruits are white, and the other ones are colored. And so as soon as fruits mature, then we can switch it over to fruit maturation. But we do have a column each for immature and mature fruits. And those two are the biggest things in the project that will tell us about fitness. And then after flowering, I already mentioned that actually. So my predictions, flowering phenology will be sooner in the northern latitudes, like I mentioned earlier, because of that short growing season. Plants from lower latitudes will have lower fitness since they're used to that less precipitation and warmer temperatures. And then pollinators will visit northern populations more often than southern populations. It'll be interesting to see, they mentioned this, that sometimes pollinators will look for certain flowers that come up at a certain time. So if bees are expecting American bellflowers to be up earlier, then they may select for a more northern population that flowers earlier. And the southern populations won't get any pollination, which may affect their fitness greatly. So that's it. Any questions?
Cool.
You find to take the seeds and try and germinate them to see fitness?
Actually, for my project, I'm not. Holly has another project, a hybrid selection project, that we actually planted seeds. We didn't plant actual plants. And those have germinated. We took germination data down for her. So we are going to harvest some of the seeds from each experiment and then grow those again for if we want to do it next year, if we want to just see how they grow. So.
Is this part of a long-term study, or is this?
Yeah, it's been going on for, I'm not sure exactly how many years, but it is not just this year. temperature sensors out there. We don't have any, they used to have cups in the ground, and they try to measure that water that way, it didn't work so well. But there is temperature sensors out there, so we can at least see, and they're at every single site all over the United States, so we can see at least temperature differences, and try to draw conclusions from that as well.
Now, it seems like all of your plants were probably of the same genetic stock, as they all originated from one location. Are you looking at all of local genetic adaptations? Are you comparing the native genetic stock to Michigan and Florida?
Yeah, actually, let me see. One of the projects, or one of the experiments that Polly's doing is hybrid selection, so they crossed a couple different populations. I'm trying to find a picture that will show. So right here, these plants are all, at least the one on the top left is Florida, and you can see the one here. This one is Michigan. So you can see like the complete difference. These are a lot more razor leaves, and these are a lot more low. But they're not crossed at all or anything like that with the native populations out here. They're just they were grown from seed from Michigan plants that they had in Virginia. That they grew them in Virginia, and we all brought them all here. So,
Okay, so they did take local genetics back to
yeah,
start
for each location.
Yeah.
Yeah.
Cool.